Namaste students. I am JV Dr. Aziz Mohammed Khan from Department of Food and Biotechnology, Faculty of Agriculture and Veterinary Science, Jyoti Vidyapith Women's University, Jaipur. Today I am going to discuss about the topic antigen. What is the antigen and what are the properties of an antigen? So antigen, the term antigen and the immunogen are the two terms which are often used synonymously. First, we need to understand what is the immunogen. Then we'll definitely understand what is the antigen. Immunogen. So a foreign particle, okay, a foreign particle, self or non-self. Some particles which are present in the human immune system, human body are known as the self, and some parts which are not present in the immune system or human body, they are when they enter, they are considered as a non-self. So self and non-self will decide what is the antigen, what is the immunogen. So when a foreign particle or a non-self molecule will enter into the body and elicit an immune response, then this type of molecule is known as the immunogen. Okay, molecule, the parts which will elicit an immune response is known as the immunogen. So they will activate the immune system. So they are known as the immunogen. And what is the antigen? So it is also immunogen, but the term is a little bit different. Antigen is a molecule, a foreign particle, which enhance the activation and production of the antibodies, which lead to the production of antibodies and which will lead to the activation of T helper cells for antibody productions are known as the antigens. So that is the different terms which are used as the antigen and the immunogen. So some another topic, one another term which is hapten. What is the hapten? Some molecules are very small in nature and they cannot uh, produce any kind of immune response, such as dinitrophenide, which are not immunogenic in themselves. But when they will react to any antibody, they will make it a hapten. They will make a hapten molecule. When they combine in another molecule, they will make a another molecule, and they are considered as a hapten. Okay, antigens are recognized not only by antibodies but are also by antigen-specific T cell receptors. In contrast to the immunoglobulins, which usually recognize intact antigen, T cell surface receptor recognize process antigen. And some another molecule which are involved in the identification of these antigens are known as the major histocompatibility complex. So major histocompatibility complex class 1 and class 2 identify these antigens. Now antigenic determinants. What are the antigenic determinants? The part of the antibody molecule which contacts the antigen is termed as the paratope. The part of the antibody which will bind to the antigen is known as the paratope. And the part of the antigen which will bind to the antibody is known as the epitope. And this epitope will decide the structure which is known as the antigenic determinant, because this by this antigenic determinant, by this epitope, the antibody, specific antibody will bind. So which kind of epitopes can be there? What are the epitopes can be there? when we talk about the structure of the bacterial cell, then there are various parts in the structure of bacterial cell. The first one is capsule, the second one is cell wall, capsid, cilia, flagella, membrane proteins, carbohydrates, mucopeptides, okay, cytoplasm and cytoplasmic cellular organelles. So these are present in the um, bacterial cells. Suppose after on the glycocalyx, when we talk about the glycocalyx, the outer side of the bacterial cell is also known as the antigenic determinant. The capsule is also antigenic determinant. The cell wall protein are also the antigenic determinants. The carbohydrates of the uh, bacterial cell are also the antigenic determinants. The cellular compartments or cellular proteins are also the antigenic determinants. The DNA, the RNA are also the antigenic determinants. So these all are the antigenic determinants by which the antigen is identified. So a antigenic determinants will decide the antigen to be identified. So what are the requirement of immunogenicity which need to be identified? How, what are the properties, what properties should be there in an antigen to show the immunogenicity? The first first thing, the antigen must be foreign. The antigen must be foreign. The antigen must be genetically foreign to the individual host. If they are same, if the antigen is same in the as the particular individual, then they will not be identified. The antigen must be different from the host organism. It must be non-self. It must be genetically diverse. It must be genetically foreign. So if it is a genetically foreign, it will be identified by the many molecules such as the toll-like receptors. They will be identified by the toll-like receptors. The second thing, the molecular size. The molecular size of the antigen will determine that how the antigen is immunogenic in nature. The molecular weight is, should be higher, should be larger. The molecular weight must be larger than the 10,000 kilodalton. Because if they are smaller in the size, they cannot be identified by the host immune system. So the antigen must be larger in size. The next is the immunogenic molecule or antigenic molecule must show some degree of the chemical complexity. It must be some chemically complex. Suppose that there are some protein molecule, they will be are identified because they are proteins and DNA and another molecule are not protein and antibodies are also the proteins. So these antibodies will identify the another proteins easily. But when we talk about the lipids, they are not the genetically diverse. They are not chemically complex. They are not complex. So they must be complex. If the lipids will bind to another molecule, such as the proteins, they are bind to the carbohydrates. They are chemically complex. So now they can be identified in the immune system. But if they are not identified, it means that they are not chemically complex. So for the lipids, it is required that they will bind to another molecule such as the proteins, such as the um, carbohydrates. So they must bind to these molecules. The next thing is the conformation of the molecule, conformation of the antigen. If the conformation of the antigen is good, then they can be identified. For example, the lysozyme molecule is a good antigen, and its native form, which consists of several amino acids. Okay, several amino acids. I'm talking about the protein molecules. Lysozyme is a protein molecule, so lysozyme is a good immunogen because it has good several amino acids. But when the sequence of amino acids will disturb, okay, it's disturbed, then it will not be a good immunogen. Okay, so the conformation of the amino acid is also important because if they are in a good sequence, they will be identified. But if they are not in a good sequence, they will not be identified. Similarly, the amino acid sequence is also important in the identification of the molecule. The another thing is that the mobility of a segment of the antigen molecule influence its antigenicity. Okay, the more the mobile, the more it will be immunogenic. Similarly, one more thing, the part of the antigen. For example, if the immunogenic part is here, okay, okay, the immunogenic part is in the upper side, okay, but if the immunogenic part is in the backbone, then they cannot be identified. If the immunogenic part is on the upper side, then they can be identified easily. But if they are on the backbone, then they cannot be identified easily. So the sequence immunogenic parts accessibility is very important, by which they can be identified easily. Antigen and antibodies will interact to each other in a special manner, like a lock and key model. If a key will fits into the particular lock, similarly, a antibody will be specific for a particular kind of antigen, like a lock. Like a lock, it will bind to a particular kind of antigen, not to any other molecule. So it is just like a lock and key molecule. And what are the forces uh, which are in the antigen molecules? The forces that bind antigen to antibody become larger as intracellular distances. These forces are in essence and have no difference from the non-specific molecules. So what are the bonds are there? The first one is electrostatic bonds. The attraction between oppositively charged ionic group is known as the electrostatic. So electrostatic bonds can be there between the antigen and antibody. Similarly, the next is hydrogen bond. Hydrogen bond is a very simple bond, and it reversible hydrogen bonds between the hydrophilic molecules will be there, and antigen and antibody may have hydrogen bond. Hydrophobic interaction will be there, similar to the manner in which the droplets of water keeps it. They are also they also form some hydrophobic interactions. This lead to the formation of hydrophobic interaction, and antigen antibody will interact to each other by these hydrophobic interaction, and it will occurs between the hydrophobic non hydrophobic region of the two molecules. The next is van der Waals forces. These are the weak forces. Interaction between the electrons in the external orbits of two different micromolecules is van der Waals forces. So these are the forces which come in the two molecules, which are antigen and the antibodies. So By using this lecture, the students will be able to understand what is the antigen, what is the immunogen, what is hapten, what is the carrier molecule, what is the properties of the antigenic determinants, what are the epitopes, what is the paratope, what are the properties of a antigen to being a good immunogen, what is the binding between the antigen antibody, what is the specificity between the antigen and antibody binding, what are the forces between the antigen and antibody, and which kind of questions can be arise. The objective questions are already updated on the University Jyoti Vidyapeeth Women University Jaipur portal, official portal, digital version 2, and the subject questions are also there. Uh, the subject questions may include explain antigen, explain the properties of antigen to being a good immunogen, explain the forces which bind antigen to the antibody, what is the antigen and antibody interaction, and explain the antigenic determinants. What are the epitopes? Write the names of different epitopes. So students can give the name of different kinds of epitope if they listen the lecture very carefully. So the lecture is over here. Thank you.